So thank you very much for the kind introduction. Thank you everyone for coming to this talk. We are Hawk Biosystems. We are a biotech company that we were originally a spin-off from the Francis Crick Institute and Cancer Research UK. And today I'm going to be presenting our technology, which is QFPro. I'll give you an overview of the principles of the technology, then an applicability of in a clinical domain, and finally some research-based examples. So our goal at Hawk is to provide researchers with the tools needed to use protein functionality in fixed tissue samples, such as FFPE patient tissue samples. So we know that protein functional states drive diseases progression and often provide the therapeutical targets. So that's why we have developed QFPro, a technology which brings together the protein spatial localization of these events, of course, the protein-protein interaction profile. So we can be looking at receptor-ligand interaction, receptor heterodimerization, immunocheckpoints, and so on. And of course, we can also quantify and identify post-translational modifications. So QFPro stands for Quantifying Functions in Proteins. It is basically an enhanced and simplified Fred-Flynn bioimaging technique. So it is a two-site assay where we use two primary specific antibodies. If we get the example of a protein-protein interaction, we're going to label protein A with a specific primary antibody and then the protein B with another one. Then we have our secondary fragments, which is one is conjugated to the donor fluorophore and the other one to the acceptor. So because we implement the principles of Fred, whenever these fluorophores are within 10 nanometers of distance and there is an overlap between their spectrums, there's going to be an energy transfer from the donor to the acceptor. And this event, we're going to be able to quantify it, and it's what we call QFPro. So if we look at these representative images, we have TIGIT labeled in green and its partner CD155 in red, and only those places where the interaction is actually taking place is where we are looking at these pink pixels. And apart from this nice spatial image, we also get a quantitative score of the interaction. So Fred-Flynn has been in the field of research for many, many years, but we have enhanced it by amplifying the signal of the acceptor. And in this way, we have overcome the problem with the autofluorescence in fixed tissue. Also, it's a very specific assay since it's a two-site assay, so we are kind of immune for false positive results. Due to the spectroscopic nature of the assay, where we are measuring the lifetime of the fluorophore and not the intensity of it, we have a high dynamic range and we get robust and reproducible quantitative data. Of course, we can spatially map these events, and since it is a two-site assay, it's very easily integrated in any laboratory or clinical workflow. So as a comparative diagram, you can see that the working distance of Fred is 1 to 10 nanometers. This allows us for the detection of specific protein-protein interactions and not just proximity events. So due to the spectroscopic nature of the assay, it allows to have a high sensitivity and high detection range. So we have developed our end-to-end solution so everyone can use QFPro. Starting from the beginning, we have our reagent kits, which is suited to any type of protein interaction that you're interested in. It has all the secondary reagents, quenches, buffers, multimedia, and so on. You just need to select your primary antibodies, and then you're good to go. We normally stain in FFPE tissue samples, but of course you can also use fixed cell lines. Then to be able to measure these Fred events, you specifically need a Flynn microscope. We developed Violet so that it can be integrated in the laboratory, because if you have seen any Flynn device, they're huge, they take a whole table, they need the dark room, air cooling system, and so on. So we try to overcome all those limitations, and we developed Violet to be a benchtop solution. And finally, we also developed the software analytics. So that's a kind of user-friendly software where you can analyze the data, and then you would get the image exported and the QFPro score. So we believe that this technology is opening a whole new family of breakthrough biomarkers, because we're not just looking at expression, we're looking at functionality. This means protein-protein interaction, post-translational modification, protein DNA, and so on. So we believe that we can get this technology within many domains, since fundamental research to drug development or the clinical diagnostics. And with this, I come to the clinical validation that we've performed with QFPro. And we did this in the immuno-oncology domain, where we know that only between 12 to 20% of solid tumor patients are responding to immuno-oncology therapies. So there's an unmet clinical need where we have to effectively predict which patients are going to respond to these type of therapies. So in a cohort of non-small cell lung cancer patients, we implemented our QFPro technology. It's important to say that all these patients were treated either with PD-1 or PD-L1 therapies independently on their PD-L1 status due to compassionate use or expanded access and so on. So we wanted to predict this patient response by implementing our assay. These are some representative images that you can see how we could label PD-1 in green, PD-L1 in red, and only those cells that are actually interacting is where you're going to see these spots. So right now, whenever a patient comes into the clinic, they will get their biopsy done. And right now, the gold standard is to be looking at PD-L1 by immunohistochemistry, and that will give you the TPS score. If we look at this Kaplan-Meier survival curve, we know that these patients that are stratified by PD-L1 expression levels are not being stratified adequately. So high PD-L1 expression does not correlate to respond to the treatment. But we thought, okay, these immune checkpoint inhibitors are actually targeting the interaction. Why don't we quantify the interaction? And we did that with our QFPro assay. And we could see that QFPro provides a significant predictive value in these patients. So patients with a high interaction level responded significantly better compared to the ones with lower states. And critically, because we got a cohort of 188 patients, we could generate this cutoff value. So 2.1% was the QFPro value. All those patients above that number would have to go to immunotherapy. Those underneath that value would have to go to other type of therapies. And if we expand these results a bit further and we plot the patients based on their PD-L1 expression levels or their QFPro score, that would be the interaction level, we can see that nearly 22.5% of patients are being treated inadequately. So PD-L1 is not a good biomarker for patient response. And on the other hand, if we look at these other 24.5% of patients, which right now are not being treated, they clearly respond to this therapy. So this is showing the need that we have for these novel biomarkers. So at the end, more than 47% of patients are being diagnosed with lung cancer and not being treated adequately. The conclusions of our study that we published in the Journal of Clinical Oncology is that if we were to implement QFPro as a predictive biomarker, we could triple the efficacy of immunocheckpoint inhibitor therapies such as Pembro or Nivo, and also the survival rates of these patients could potentially double. We did then another study with another cohort of patients. In this case, they were also non-small cell lung cancer patients. Again, they were all treated with anti-PD-1 or PD-L1 therapies. And in this case, we look at two different biomarkers. So we stain for PD-1, PD-L1, and for CD8, CTLA4. And what we could observe is that those patients who had high interaction state for both immunocheckpoints had a significantly higher response to treatment than those ones that showed low interaction levels of these immunocheckpoints. We could also observe these results in overall survival and progression-free survival. So these results show the value that QFPro can have in helping treating decision-making for immunocheckpoint inhibitor therapies, and also the potential to identify novel functional biomarkers into the clinical domain. So we, of course, it's not just about lung cancer and not just PD-1, PD-L1, or CTLA4. We are aiming to generate a whole immune profile in all the immunogenic tumors. So that way, each patient could receive their specific therapy and the one that they would respond to. And in this case, we have an example that within a single FFPE tissue of a patient, we did 3 different rounds of staining within this same tissue. First, we look at PD-1, PD-L1 interaction. Then in the second round, we stain for CTLA4, CD80, and finally, LAG3, MHC2. This gave us a profile of the interaction status of this patient within these 3 different biomarkers. And hopefully in the future, this patient will receive maybe anti-LAG3 treatment instead of anti-PD-1. And now I just want to briefly show you some other research-based applications that we have developed in the laboratory, so you have a little bit more of an idea of what can be looked with this type of technology. So in this first example, we look at the intracellular interaction between E-cadherin and beta-catenin. Therefore, we stain with their primary specific antibodies, then we use our secondary fragments, we perform the QF-Pro assay. And this was done in FFPE tissue of breast cancer patients. As you can see, both patients express both beta-catenin and E-cadherin, but only patient number 2, where we see these red pixels, is where the two proteins are actually interacting. And where our real strength lies in is in the quantitative value. So we can see that the patient number 2 is the one that is giving you this interaction state. If we were to overlay patient number 1 images, and we will to merge them, we would imply like, okay, they're co-localizing, but actually they are not interacting, and this is very important. Another example that we did was in cell lines. We did at the interaction between TIGIT and CD155. And in this case, we wanted to treat the cells with an inhibitory, a blocking antibody, to see if we could disrupt this interaction. And as you can see in the representative images, we lose the signal, the red pixels. And again, it's the quantitative data that makes it real strong. And a final example is in the post-translational modification domain. In this case, we wanted to study the activation state of AKT or PKB. This way, we label with one primary antibody the whole protein and then another antibody for the residue threonine 308. So in these cell lines, what we did was to treat them at different time points with an inducer, so epidermal growth factor, that would induce the activation of AKT. And as you can see, we get this nice time response curve. Upon higher treatment, we get a higher activation. And of course, you can see how in the images, the phenotype is switching from a bluish kind of blue color to a more red and orange phenotype. So we've got a whole family of biomarkers that we have validated in our laboratory. So we normally say that anything that can be targeted with 2 primary antibodies is subjected to be a QF-Pro assay. We have some other that are not disclosable for our partners, but of course, we are always happy to find novel biomarkers and to collaborate with any researcher. So I'm also very happy to say that we have an exclusive distributor here in the Netherlands and Belgium, which is BioSPX, which we're sharing our booth here. And they're the ones that know most about the technology here. So just to let you know how we can work together, we've got our product portfolio. So what I said, the reagent kit, film microscope, and the software. Also, we provide service as a test, so you can send us your sample to our laboratory. We will do all the staining acquisition and then give you back with our report. And for pharma companies, we also provide companion diagnostics. So with that, I think I've finished my presentation.